We are moving forward with a new selected oral presentation from Davide Ferraro, who is coming from the University of Padova, and he will speak about fast and reproducible methods for extracellular vesicle isolation by novel droplet microfluidics device. You have.
Thank you. So thank you for the introduction, and thank you for the organizer for accepting the presentation here. So yes, today I want to talk about the droplet microfluidic device that we developed for extracellular vesicle isolation. As you will see at the end, this is a very interdisciplinary project that involves a lot of people. So I don't think I need to introduce extracellular vesicles anymore. I think the key point about their use for liquid biopsy is clear now in the audience. However, there is 2 points that I want to stress out. That there are still a couple of pivotal aspects that must be faced before using extracellular vesicles for example, clinical application. And the first one is the purification method, because there is no one purification method that works perfectly for all the application, but everybody has different strategies, basically, and if you face for the first time extracellular vesicle world, you are a bit puzzled. And the second one is the heterogeneity, because it is some kind of a young research field. We don't know so much about extracellular vesicles, and especially the heterogeneity. We know that one single cell can release a lot of extracellular vesicle with different cargoes, but we don't know exactly what is inside of every vis, for example. And we think that microfluidic can support both these needs. And today I will discuss about the first point, which is the easiest part, let's say, that is the purification method. So for the people who are not familiar, there are mainly 2 methods for isolate extracellular vesicle, or EVs. The first one is sequential ultracentrifugation, basically, where you precipitate a sort of pellet that you collect and then you analyze. This process works. It's probably the most used worldwide process. The problem is that it's not completely reproducible between different labs because it's very operator dependent, basically, and is prone to contamination because together with EVs you collect protein, lipids, and a lot of other things. The other approach is the size exclusion chromatography, as we have seen before, that, however, is very labor intense because it's quite long. Before performing seq, you have to perform anyway the ultracentrifugation, otherwise you have too much sample to be analyzed. And then now there are people starting thinking that actually seq provide an too high purification, because basically you are removing apparently part of the corona that was discussed before around the EVs, and these may have a physiological rule. So there is a lot of discussion behind that. Recently, people start to use the membrane, the protein membrane that are around the extracellular vesicle for affinity capture, because it's known that all EVs basically has a specific protein, in particular tetraspanin, and so we can use microarray or magnetic beads in order to collect them, for example. However, the procedure typically are quite long, expensive, and still operator dependent. So what we try to do is to use microfluidics in order to face this problem. So for people who are not familiar with, microfluidics was born roughly 25 years ago with the idea of integrating inside of a microfluidic device all the typical lab scale approach that you have in a chemical or biological protocol. And this, of course, would lead to a lot of advantages, as you can see here, with this concept of lab on a chip. Then, roughly 15 years ago, was born a branch of microfluidics that is known as Droplets microfluidics. Actually, we have seen yesterday a lot of applications that are commercially available about droplet microfluidics. But the main idea here is that we want to create an emulsion. So if you take oil and water, you shake, you create an emulsion. However, if you look at your emulsion, all droplets has different size and they are not stable. Thanks to microfluidics, you can perform very stable emulsion and very controlled emulsion. And thanks to the research of the past years, now in the literature you can find whatever protocol using droplets. You can break the droplet, you can merge the droplet, you can even manipulate magnetic beads inside of the droplet. Here you have basically a magnetic tweezer, you extract and you release magnetic beads from from the droplet. These, of course, increase the advantages of conventional microfluidics, and the main two key points, at least from my point of view, is that every droplet can be considered as a single independent reactor from the other droplets and from the channel wall. So in principle, the devices are not disposable. And then inside of a droplet, you have a rapid mixing of whatever you mix inside due to turbulence that are automatically generated while the droplet is flowing. And these led to a lot of applications, and some of them are here, and some of them were mentioned yesterday. And there are others actually that are based on droplet microfluidics. So if we match the board about microfluidics and extracellular vesicle, people basically tried so far to isolate extracellular vesicle mainly by affinity capture. So the first example were pretty interesting and relatively simple because people took a microfluidic device, coat with specific antibody the microfluidic device in order to catch the EVs. This approach was working, but was of course limited to the bonding probability between the molecules and the extracellular vesicle, and so were a sort of proof of concept that was working just for microliter. Then filtration is also integrated in microfluidic device. These are working pretty fine in terms of throughput. The limitation of this approach is that in order to realize these nanopillars, you required expensive facilities and trained people, and this device being based on filtration, they are disposable. So the scale up in a real application is complicated. And then recently people start to put inside the microfluidic device magnetic beads. So you can imagine here you have your sample, here you have your beads, you co-flow the two together, and at a certain point you have your beads decorated with EVs. The problem of this approach is still the sample throughput, which is very limited. And the main reason here is actually the microfluidics itself. Because in microfluidics, you have laminar flow. So when you mix two liquid, after, for example, 10 centimeters, the two liquid is not mixed yet because the two flow in parallel, and so the mixing is just at the interface. There are strategies to increase the mixing, but actually the best option is using droplets. Because using droplets, you can basically mix the two liquids after a few milliseconds, and you have a complete mixing after a few milliseconds. This is known since more than 10 years, basically, in droplet microfluidics. So based on the idea, we choose to use droplet microfluidics for isolate extracellular vesicle, and we develop a platform that is able at first to generate droplet. This is a typical droplet microfluidics generator, where you have droplets spaced by another oil that acts just as a spacer. And inside of every droplet, we have beads and hypothetically EVs. Then we have this droplet can be incubated for a certain time and at a certain temperature. It's important to mention here that basically to handle, for the droplet generation, to handle 200 microliter of sample, we takes 1 minute. And we tested up to 2 ml, and this takes 10 minutes, basically. Then the incubation can be triggered as desired, and then finally, we can collect the beads outside the droplet using a system that I showed you at the beginning before, similarly, where you have a magnet here that extracts the magnetic beads from the droplet. Here you see a small cluster that is generated. And again, these take 5 minutes for 200 microliter and 20 minutes for 2 ml. Then in order to validate our platform, we designed 2 parallel experiments using commercially available magnetic beads coated with CD81, which is one of the typical markers for extracellular vesicle. One is the workflow proposed by Thermo Fisher that basically consists in mixing everything together and then wait an overnight incubation on the thermomixer. This is the idea. And the other one using our microfluidic device. So after performing experiment, we characterize at first the quantity of protein we can extract from the magnetic beads, with the idea that these proteins are coming from the extracellular vesicle. And this, for that we were very happy because basically you can see that after 4 hours of incubation inside the microfluidic device, we have much more protein than using the in-batch incubation. And this is just probably because inside of the droplet we have an enhanced mixing with respect with the conventional thermomixer, just because with the thermomixer you're not mixing the beads. The beads are stay at the bottom and you mix the solution basically because the gravity is too high. And then the other point is that, okay, we perform some Western blot in order to characterize our protein, and we check just that we have there actually the protein we expected from extracellular vesicles, so CD9, CD81, CD29. Then we use alkylexing as a negative control, but actually we're not present even in the starting sample, so it was not a real negative control at that moment. Then we perform some characterization in terms of extraction efficiency, so we use the conventional NTA approach. In that case, we analyze the starting sample, then the supernatant after the microfluidics, and this is a critical point because at the moment we are not able to detach the EV after having captured on the magnetic beads, so we analyze the residual supernatant, and according to that we have an extraction efficiency of about 56%, which is reasonable for affinity capture, because you have to think that not all the EVs express the CD81 that we have at the beginning probably, and so it's normal that you have a subpopulation. If you compare other affinity capture approach, typically you range between 30 and 70%. So probably we catch all we could catch at least using this approach. And then we try to understand if our EVs were actually intact after having capture on the magnetic beads. And for that we perform some flow-cytometry using EVs made fluorescent actually. And in this case, this is not nanofax for the people using extracellular vesicle. This is conventional fax because our EVs, remember, that are attached on the magnetic beads. And so what we see there are basically the magnetic beads that apparently are a bit fluorescent due to the decoration given by the extracellular vesicle. And then we perform some confocal microscopy where this is the beads basically on the white field, and these spots there are probably aggregation of extracellular vesicle over the magnetic beads. And then finally, we characterize the microRNA extracted from the extracellular vesicle isolated by the microfluidic device with the idea, okay, if we can catch the microRNA after the old protocol, it means that our extracellular vesicles are still intact. And we perform a comparison between the microfluidic setup and the in-batch approach, finding more or less the same results, so we were quite happy about that. So in conclusion, I hope that I convince you that droplet microfluidics can be a possible solution for extracellular vesicle isolation, and that here we provide a relatively fast protocol because in 4 hours and a half we could achieve at least the same results, even if slightly better, than an in-batch approach that takes between 12, 15 hours. And actually what is important here is that also we can work in the same time frame with 2 ml of sample, while the typical protocol are designed for hundreds of microliters, let's say. And this is just because of droplet microfluidics, because the droplet generation takes few minutes, and then the incubation can take between 1 hour or 10 hours. It doesn't matter because beads doesn't escape the droplet, so we can keep flowing as much as we want. Then we have an isolation of about 56%, and this is apparently reasonable. What we are doing at the moment, so we are working to, of course, increase the throughput. This is not anymore microfluidic problem, it's an antigen-antibody reaction optimization, basically, because this 4-hour incubation depends on the beads. So we are using different beads. We are working with collaborators designing basically beads. Then we are using now human plasma as starting sample, because from the microfluidics point of view, this can create some problem because plasma, for example, is not a Newtonian fluid. For droplet generation, is not that obvious. And then we have a project in collaboration with the chemistry department of the CNR in Milan for developing magnetic beads where we can eventually detach this extracellular vesicle from the beads itself. And then what we want to do in the late future is to study the heterogeneity of extracellular vesicle, because we are convinced that droplet microfluidics could be a good tool also for investigated, coupled of course with sequencing, for example, for investigate extracellular vesicle heterogeneity. So to conclude, I want to thank all the group where I belong, the LAFSI group in the physics department, and in particular Alessio, who is here in the audience, that did most of the work that I presented here. And then the group of Valentina Zappulli from the BCA department of the University of Padova, that are very expert on the extracellular vesicle, Professor Paola Brun from the Department of Medicine, and finally the team of Lara Mussolin that support us especially for the microRNAs isolation. And then, of course, the grant that support this activity during the past 2 years. And thank you for the attention.
Thanks a lot, Davide, for this very interesting presentation. Maybe we have time for one question.
One quick question: how do we determine 100% when we say 56% is efficiency?
We just subtract the initial, we characterize the initial sample by NTA, and then we characterize the supernatant, and then we subtract the two, and we hypothesize that all that we miss is attached on the beads. Actually, it can be wherever. It can be attached to the droplet, it can be lost somewhere else. Assuming also that. I agree that NTA alone, I wouldn't trust. Combined with the Western blot and the other strategy, I'm almost convinced that there is also a problem with dilution about the NTA, because you need to dilute a lot, and so yes, I agree.
Do you have an idea how many vesicles are in one droplet?
No, we start from a sample that is 10 to the power 10 per ml, and the hypothesis that is equally distributed within every droplet. And we generate droplet which is 1 microliter, a bit less. I have to do the calculation, so this is the hypothesis. What is curious is that from the facts and even from the confocal microscopy, apparently most of the beads are not decorated with the EVs. And doing the calculation, this doesn't work, doesn't match. So, or we are losing EVs somewhere else, but we are not, this could be possible, but in that case, the Western blot wouldn't be so good in terms of goodness of Western blot. The other hypothesis is that the fluorescence is not high enough. I don't know.
Thank you. Okay, so thank you, David.
Thank you.